guess, for anybody in the back row, but that would only really be Marco. Unless I go over here, then it would be Miranda. But if you can't see me, you can watch the video. But it's not going to track me and follow me around. I have a little device that the swivel follows around. It's really kind of cool, a little robot. But I didn't get a chance to play with it because I didn't come up here very early. So I wanted to do two things. Well, let's talk about grades. So your midterm grade, and I don't know what they do with high school students. I gave some of the grades to Miss Brunson. Brunson? I think it's Brunson, but I'm old, so I'm thinking Charles Bronson. But here's what I did. I averaged your 2 tests, and we only have 2 tests, and we're going to have at least 3 more. So, and the other thing is, we haven't gotten any lab grades yet for sure. So I just gave everybody an A in the lab. The lab counts 25%, and I averaged that. Okay. The grades on the last test were not very good. In fact, the average was below a 70. I haven't curved it, but what I did do was instead of taking, there were 25 questions, 4 points a question. In the last test, there were 30 questions, so I just basically took off 3 points because I don't like working with thirds. Nobody should have to work with 3s or 8s, you know. So, 2s and 4s and 5s, I'm good. But 3s and 8s just complicate things. So those questions on this last test, I just went ahead and took off 3 points each instead of 4 points. So that's why your grades may not, the math may not automatically make sense, but it makes sense to me. It just was kind of a built-in curve. The first 2 tests are always the hardest, the next one's over cells, chapter 4 and 5. But nothing from chapter 5 that isn't already in chapter 4 on the membrane part. The membrane function and transport and components of the membrane, if you look back in 4, it talks about how the membrane is a lipid bilayer and made up of proteins and lipids together, and cholesterol controls its, its fluidity.
Here's what I found from Britannica. Membranes consist largely of a lipid bilayer.
Well, that's really handy. Thank you. I don't know why. I hate my new iPhone. It's that edge to edge, and if you just barely touch it, it takes you somewhere you don't want to go. So.
And if you have like your Siri on, like it.
Well, Siri's always listening.
Yeah.
Or Alexa, or somebody is always listening.
You can turn it off.
Yeah, I can turn her off, but I use her a lot actually.
Yeah.
But, you know, I have a friend who was just telling me yesterday, he mentioned his wife's birthday is coming up and it's still cold, or it was cold in Texas last week, and he said, maybe I'll buy her a cardigan. And I said, you can sleep on my couch when you give your wife a sweater for her birthday. But he just mentioned cardigan, you know, not sweater, but cardigan. And all of a sudden, he's getting ads everywhere for it. And he didn't say, hey, Siri, look up cardigans, or hey, Alexa, shop for cardigans. He just mentioned it to me on the phone. Hi.
May I see Rhino for a minute?
Yes. Yes. So, your lab counts 25%, so I multiplied your test average by 75.75 and averaged in an A, because, you know, we're going to do the lab reports together. So if you just do them and hand them in, you know, we're going to go over them and discuss them together. Everybody makes an A or B in the lab, right? So I just averaged in 100 for everybody for that. So that's where your grade came from. But like I say, these first 2 tests are the hardest. The next one's going to be over cells. I want to stop at the end of class today and go back over and refresh it and tell you what I think is important and how to study. What else? That's it about grades. Yeah. Chemistry was much harder because they've got like 11 assignments, and some of them were just to do Monday. And this is what they look like. So these are not easy to grade when you got 30 in one class and 8 in the other. So they got their grades based on 6 assignments and like 1 test, and it was a very clear split. They get it or they don't. You know, it's like half A's. I wish I could say the other half were B's though. So the, you know, I just put in the grade that they had at that point, but this is not really even the halfway point when you really think about it. Oh, did my computer go to sleep? Was it an activity? That's not good. Okay, we're back up. So, I want to make some comments about Blackboard. Well, that's interesting. I touched it just the right place when I turned it back on. So here's the video from last week. But like I say, it kept turning off. And I don't know if the video, there was somebody in here yesterday when I came up to work and put in grades, and he obviously didn't want anybody listening. Or not. Or not. So this is, if you miss something, I'm gonna be in here. You know, I'm gonna be posting these and trying to get them done. And
So we,
If
ourselves,
I
our
can.
bodies are 40 plus.
goodbye. So, so grades, we'll talk about the test after this. This is there. Okay, so I need to talk about photosynthesis, and I'm sorry, I left my jump drive here. And when I went to print out my little friendly handout that I like to give you so you don't have to take notes yesterday, she said she wanted to borrow Rena for a minute. Well, that was not truth in advertising. Because I guess you can watch the video. So,
I like the idea of this one.
something that took more than a minute.
Yeah.
So, let's see if I can do what I usually do. Because I'm not sure where my let me get out my phone. I've had a lot of trouble with this two-factor identification too. Don't call me. I just moved the mouse a little bit. Here it comes.
Sign-in system. If you are trying to sign in,
Hit pound,
press
right?
the pound key to finish
Yeah.
signing in. If you are not trying to, your sign-in was successfully verified. Goodbye.
Organization what? Needs more information for something?
I just have it text me.
Yeah, I do too.
Yeah.
Yes, I want a text. Okay, don't change anything. Yes, it's up to date. Okay, so biology. And I was able to, rather than at my YouTube page, YouTube video page, post that Wednesday night, and I'll post tonight next time. But I did post several things, so that to me, is this gonna download them or open them? It's gonna download them, okay. And here's another one. And here's another one. And oh, I accidentally uploaded one twice, okay. You know, it didn't load real fast right the first time, and then it did. Okay, so I have 2 PowerPoints. One is, well, they're both quite good. They're just from the publisher, but some teachers have played with them. obviously change the background and everything, and move things around, and, you know, kind of change things, and added some information about separate plants. But one of them I thought was the actual one from the publisher. If you look, you can see it here on energy metabolism and photosynthesis, and see that this is from Sylvia Mader, 2018, and a somewhat simplified version, but not necessarily in the exact order from your book, and it's a little simplified, so we're gonna go with this one. But if you go there once again, if I can get back to here, you will see, that's a PowerPoint. This is the chapter outline. Here comes Microsoft Word. Here's some sample questions. And I don't know why that one didn't come up. There are lots of resources, is my point. And you know, these are the generic learning objectives and things that I plan to cover tonight, and except for the other types of photosynthesis. We're going to keep it fairly simple. And the rest we're gonna go through. And for every chapter, I've been posting questions from the test bank, and you haven't been taking advantage of them because they are the exact questions for the test. Now, I'm going to, now that you figured that out, change things up a little bit, but they were there for you. And a nice outline that's probably more detailed than you want, but if you have a computer or you can get to this, you can kind of follow along with me now. So, let me go to our discussion for the day and make sure that this works. One of these. Okay, so photosynthesis is anabolic. You may have heard of anabolic steroids. Those are steroids that build you up. And photosynthesis builds things up from very small and simple molecules into much larger molecules. And it's endergonic, or, well, we don't really want to call it endothermic, although you might. It requires an input of energy. Endothermic is an input of heat energy. You have endothermic reactions. Those are ones that soak up energy in the form of thermal energy from the environment and feel cold because they're taking, when you touch them, they take heat from your finger and your nerves sense that. But gonic refers more to energy. Carbon dioxide, a very simple inorganic form of carbon in all your breath, but made not just by living organisms, but in all kinds of reactions. Some acid-base reactions, some, it's spewed with methane and other things out of the earth in different geological processes. But this process is only living, and it uses light energy, the energy of light, and water, the reducing power of water, I'll talk about that in a minute, to produce sugar. In fact, your book starts out with some incredible, I don't know, how many million or billion tons? Oh, I left your book open to the right place, I can tell you. But 200 billion metric tons of sugar, enough to recreate the volume. If you were to pile that much sugar in one place, it would have the volume of, I don't know who does these calculations, but 2 million Empire State Buildings. And if you've ever been to 34th Street or whatever, or 32nd Street, I forget what it is, just down from Grand Central Station in New York City, and been to the Empire State Building, it is very impressive. It's hardly the world's tallest building anymore, but it's very impressive, huge building. So, lots of sugar. About 1% of all the sunlight falling on the earth is absorbed by plants and used for photosynthesis. That's the energy falling on the earth. Only about, I forget how much, 10% or so, I don't know. Most of it's reflected anyway. And then of that gets in, a little less than 1% is used to make sugar. And we're calling our glucose, that sugar molecule, C6H12O6. And in chemistry, if you know about empirical formulas, this one's empirical formula is CH2O. That's the ratio. 1 carbon to 2 hydrogen to 1 oxygen, 6 times. However you want to put it, or 6 over here. So, this is where the term carbohydrate, carbon, hydros means water in Greek, comes from. And carbohydrates mean sugars. So we're going to make sugars. And then next week, we're going to eat them and break them down. And you're going to see a lot of similarities in the two balanced equations because a plant, there should be a green one for tonight. Anyway, a plant takes carbon dioxide from the air plus water from the air, from the roots, from the ground, from the sea, wherever it's, from the pond, wherever it's living, and use it to make this glucose, C6H12O6. This will be our starting point for respiration. And another big deal is, thank goodness, the plants make oxygen. This process was first observed on Earth using the energy of the sun to catalyze this reaction. And so, this particular life process has been on the Earth for about 3 billion years. Okay, there are these things called stromatolites, stromatoliths, that you find first in Australia. You can find them at Capitol National Park in Utah. And they age out to about 3 billion years ago, the Earth being about 4.5 billion years old. So, previous to that time, and we have ways of telling this, the atmosphere was highly reducing and reduced. And by that we mean full of methane and carbon dioxide and all kinds of things, but not much oxygen. And then this major pollution event took place where these photosynthetic organisms started spilling out so much oxygen to the point where today our atmosphere has about 20% oxygen, 19-20%. So photosynthesis takes place in plants, and basically they're producing their own food, and that introduces the term autotroph. I wish I had my handouts because I have this in bold print. And troph means to eat. And so these are, auto means self or self-propelled, like an automobile is self-mobile. Autotrophs are self-feeders. They produce their own food. And through several processes, there's photosynthesis, and then there are photosynthetic autotrophs that use the energy of the sun to make glucose, but there are also chemoautotrophs or chemolithoautotrophs there are some subdivisions of this, but the two broad classes are chemoautotrophs and photoautotrophs. They might live in an environment where there's no sunlight, but they use chemical processes to extract the energy that they use to carry out this same reaction. Photosynthesis, of course, occurs in the leaves, and, you know, I would have asked another question, and that is, how does the CO2 get in the leaf? The leaf doesn't have enough surface area. Remember, there are some things, some trends that are going to repeat over and over. Structure determines function, and surface to volume ratio is going to become important. And I'm going to show you how these leaves increase their surface area. I'm not teaching botany, but the structure of a leaf is such that if you pull it open, it has intricate cells and air passages and multiple layers of cells that allow for more surface area to capture more sunlight. Because, you know, trees want to spread out, and the more leaf and the more surface area you have, the more photosynthesis you can have and the more growth you can have. They come in, the CO2 comes in through tiny holes on the underside of the leaf. I have a cool experiment about that. You know, you paint the underside with like fingernail polish and you pull it off and you can see the holes. We can just look at a leaf under the microscope. Low power, because the light's not going to shine through it. Or maybe I'll bring in my dissecting microscope and we'll look at those pores. You can kind of see them even if you look closely and you know what to look for without it. And one of them is a stoma, and many are stomata. What's stomata? I don't know. What's stomata with you? So the stoma are just little pores that allow the CO2 to get in because it can't diffuse fast enough. I also have a really good lab on diffusion we're going to do. We're going to make cells. A little cell about this big, and then another one about this big, and then another one about this big, and we're going to measure how fast stuff diffuses into the center, or how far it diffuses in a certain amount of time. I'm still waiting on some things to come in. So, those cells inside the meaty tissue are called mesophyll cells, but you don't have to know any botany really. But you should know stoma, because I'm going to talk to you about something else called stroma. And the stoma are pores in the leaf surrounded by two cells that can open and close. They don't open and close on every plant, but some, for example, they close at night and open during the day. Or maybe they do the reverse. But, you know, if they close at night, they trap the CO2 in there for what we're going to call the dark reactions. That'll become important later. But these two cells are called guard cells, and they open and close by swelling up and shrinking down through osmosis and diffusion. That's why we talked about that. I have a lab on osmosis and diffusion. Let me just say, I know chapter 5 included hypertonic, hypotonic, isotonic, osmosis and diffusion. I'm not going to test next week over that. I just want the facts. Parts of the cell, their structure and their functions. We're going to do that in a lab. So I'm going to not test you over it, but I'm going to get you to study it. You know, I can make you study it by saying I'm going to test you over it and you have to go home and read about it. Or we can just do it in the lab and you can suffer and listen to me talk about it. But either way, you'll get the same amount of exposure. So that's my goal for the lab, is to fill in some of these blanks like diffusion. What does stomata look like? How does osmosis work? We're going to do it in the context of dialysis and making hypo and hypertonic solutions relative to the solution or the contents of a dialysis bag. Anyway, I should go through this. So stomata are pores in a plant's cuticle. The cuticle is the outer surface through which water vapor and gases are exchanged between the plant and the environment. On either side is a guard cell. Carbon dioxide goes in. Oxygen is a waste product in these plants, and we use it. We also use the plant and we eat them. So, this is what I mean by mesophyll. See all this meaty, spongy tissue in here with rather loose, well, it says spongy mesophyll, with rather, loose connections so that air, which contains both oxygen and CO2, can diffuse through them. Okay? And then, of course, the veins carry nutrients in both directions and water. So, this person must have been about this. But if you look at these cells, you know that plant cells have several things that animal cells don't have. A rigid cell wall, because the plants don't have any bones. So they use cellulose to hold themselves together, and then they make bundles of cells that form long fibers that pack together to form more fibers, and you end up with a tree. You build small and you build up. These photosynthetic cells, though, the ones carrying out photosynthesis, the ones with chloroplasts, that's the difference. They have a cell wall and they have chloroplasts, right? And they frequently have a large vacuole because regulating osmotic pressure is so important to them, and as is storage of the sugars and everything produced as starch. They take more room. So, here's a typical plant cell. It's got a cell wall, it's got a central vacuole, large central vacuole, like everybody. It's got a nucleus, mitochondria, endoplasmic reticulum, and Golgi. And the photosynthetic organ is the chloroplast. Okay, so you're going to get a break on the next test. Did I hand out the coloring sheets for that?
Yes.
Okay, but if you didn't get them or you weren't here, I have them. And I went ahead and did mine. I shouldn't have, because I was sitting here saying, I'll just do these while I'm waiting. And then someone came and said, do you have your grades in yet? And I'm like, no, nobody told me my grades have to be in at 4 o'clock yesterday. And I said, well, thanks for telling me. And then I went and turned your grades in real fast. I mean, I just took what grades, what grades we had recorded. Okay, so I have some extras, if I can find them. Because not all of them are double-sided, like that one, because what? Because I use those for my originals. That's one-sided. That's one-sided. Two-sided, two-sided. And one-sided. Okay, I have 2 animal cells and 3 plant cells. But I can stop and go make some more. So you need to know for sure, right? Plant and animal.
Thank you.
And were you here that day? You got plant and animal? Who wasn't here? Okay, if you don't, I have 2 more. Okay.
Yes, I have.
Okay, so 10 points. And if you do this, I gave everybody credit for doing it last time. And I know, what are you doing in biology? You go home and your significant other or your parents or something say, so what'd you do in biology? Oh, we colored. But, like I say, this is from a regular college textbook. And if you trace the letters, don't just create a color key and rub over them with your color, and don't just, I had someone, this is last year, I had someone put a little square for their color key next to them. But if you trace over the letters, you stimulate, the area of the brain called Broca's speech area. It's the speech language center of the brain, and it facilitates retention. So see how I trace the letters? I know it's time consuming, it doesn't seem to really make a difference, but it'll make a difference in how this works. And then I'm going to show you something else after we finish this to show you how to make a difference. But not only that, when you go through and read this, for example, on the plant cell, it says the plant cell membrane is essentially the same as an animal cell membrane. Immediately outside the plant cell membrane, however, is a cell wall consisting mostly of fibers of cellulose. There's like a one-page summary of everything going on in the plant, Plants are colored by their plastids, but chloroplasts, which are green, have a much more important function than merely making plants green, which we're going to talk about next. They trap light energy and convert it to chemical energy for the manufacture of food in the process called photosynthesis. And we're going to talk about that chloroplast in a little more detail soon. But you'll see, it describes, as you read in color, it says, color titles and structures A through H, including the heading plastid. Use green for F and a light color for D and D1. So, anyway, use whatever colors you want, but obviously, plant cells, my plant cell green, so we shall see. Let me put these up while I'm thinking about it, though, so I can be sure.
We don't have to use the exact colors you use.
No. You haven't done it yet, but I always have, when you ask a question like that, it reminds me of the question the person has said, do you prefer, or am I supposed to use pen or pencil? And I'll just say, I don't care, whatever you want. And they'll go, but which do you prefer? And I say, you're one of those, okay? And I just go, pen, because it's easier for me to read. And that's because women, especially, have a much more advanced dorsolateral prefrontal cortex. It's right about here. That sounds complicated, but as you'll learn in anatomy and physiology, dorsolateral, dorsal means to the back, lateral means to the side. Your prefrontal cortex, this is your frontal cortex, your prefrontal cortex is behind it, and it's right about here. And it's highly developed in some women, like my daughter, who's extremely anxious, because it's called the seat of executive function, or seat of executive functioning in the brain. And it makes you plan and think about consequences. Like, what if I use a pencil? He won't like it, or it won't work, or something, anyway. Whereas in males, it usually doesn't develop until about age 21, which the insurance companies have long known, because when does your insurance rate go down? When you turn 21, because get in a car with an 18 or 19-year-old and you might as well, put a gun to your head, because it's like, okay, you get a car, what's the first thing you do as an 18-year-old, Let's see what this baby can do. Let's see how fast I can take this curb. Let's see if I can get it airborne over the railroad tracks. He's laughing because he's experienced all of this. So, you just want to do these and trace these. This is a brain-compatible learning method. I'm going to show you one other one that will help you slow down and think about it and do it. But this involves tactile, because you're coloring and moving, and obviously the visual, okay? And then it's also, like I say, stimulating the speech language center. And when you stimulate multiple areas of the brain at once, it stimulates movement of information into long-term memory and facilitates recall. So it's just, I'll give you 10 points as an incentive, but the real incentive should be just because it's a good way to learn. So, we're going to talk about the structures of these chloroplasts in a little bit more detail. So, they are a double-membraned organelle. I talked about the double-membraned organelles. The nucleus is a double-membrane organelle, but its double membranes are so unique, we even call it the nuclear envelope. We give it a new term. But mitochondria and chloroplast originated as symbionts, living inside cells with their own, I won't say independent existence, but their own DNA, their own structures, and they've gotten degenerate to where they rely on the host cell, but they still have their own ribosomes, like bacteria, what we call 70S ribosomes instead of 80S ribosomes like the eukaryotes. They have their own DNA, it's very small, codes for their ribosomes, and some of the proteins in here. And it's circular, like bacterial DNA. So clearly these were once endosymbionts. And they're very complex, and this isn't a really good drawing because if I were to draw a chloroplast, I would draw it with its outer membrane, and its inner membrane I would draw like this. And I would connect it to the inner membrane because these flattened discs of membrane are called thylakoids. And they're stacks of inner membrane, there we go, that are invaginations or out pooches of the membrane, but multiple pooches that stack up on each other. And when they stack up together, one of them is a thylakoid, but when they stack up together, it's called a grana. They're called grana. Sorry, one's called a granum. And I am sorry, I can't give you any rules for what is plural and what is singular. Because some are Latin words, and so you have like nucleus, nuclei, and some are Greek. So you have granum for one and grana for many. So you just have to learn. Mitochondrion is Greek, and many of them are mitochondria. Although you hear people say a mitochondria. They really should say a mitochondrion. As a scientist, that bothers me. And as a Texan, the thing that bothers me is people say they want one tamale. You ever heard that? These gringos, arroz blanca around here. It's a tamal. Okay? Many of them are tamales. Anyway. So I don't have any good rules for you, but like I say, one of these flattened discs is a thylakoid. Here's that theme again. I want to increase my surface area because this is the site of photosynthesis, and the more thylakoids I have, the more photosynthesis I can do, and the more sugar I can make in a smaller amount of space. So, like I say, when they're stacked together, they're called grana, and they're all contiguous, and they're connected. They're just enfoldings or invaginations of the inner membrane. The thylakoid discs, like I say, stack into grana, and the inside of them is called the thylakoid space. That's going to become important because we mentioned it, but only briefly, that the membrane is asymmetric. The two faces of the membrane are different. And the inside and the outside of this membrane is going to become important and have important differences because we'll go into more detail with the mitochondria. We're going to do what we call a vectorial ejection of the hydrogen ions from water. When we split water into oxygen and H+, and pull that electron out to give it to the CO2, we're going to put all the H pluses on one side of the membrane, and the thylakoid membrane, and then it will what? Hello? It will create a little battery, a differential voltage, an electrochemical gradient. Anyway, so why are plants green? Because in the thylakoid membranes are two pigments. Well, there are many pigments. Chlorophyll A and chlorophyll B are the main ones, and they are very similar to heme. They're a porphyrin molecule, except they don't have iron, they have magnesium in the middle. They're energy harvesting antennas. Some absorb blue. I don't have a spectrum chart, but one's coming up, and some absorb red. And that's why plants are green, because they absorb red and they absorb blue, and they capture it. But they don't absorb the green. It reflects and reflects back to your eyes, and you see them as green. So that's why plants are green. If you look at the absorption spectrum, if I look at how much energy is absorbed, that's absorbance. And this is my wavelength of light. Color of light, and it's gonna go, let's go from high energy to low energy. So let's go from low energy to high energy. So I'm gonna have red down here, and blue and violet up here. And of course, this would become red, orange, yellow, green, blue, indigo, violet. Blue up here, and then violet up here. Colors of the rainbow. And it absorbs very highly here, falls down in the green, and then goes back up in the blue. So, for those of you growing, I hope, African violets at home, and using plant grow lights, when you buy a plant grow light, it's shifted so that you have more red and more blue, and less green. In a minute, I'll tell you how to save money. Anybody who's into fashion or performing or makeup knows you can buy makeup mirrors with a little switch on them that switches from daylight to sunlight to stadium lights to stage lights to fluorescent lights, office lights, because these all produce different colors. You can look up and see some of them are yellow, some of them are white, and you can buy them with different colors in them, with more yellow in them or more blue in them, etc. And that's the way, those grow lights work, is they emphasize the red and the blue. But I'll tell you right now, the normal fluorescent lights and normal lights you buy, you don't have to pay 2 or 3 times as much. The normal fluorescent lights you buy still produce plenty in the entire spectrum, so you don't have to spend extra on grow lights. There's a good science fair experiment in there, but I'll tell you what everybody else has found. The grow lights don't make a difference. They'll say, the lights, when you look at the light package, it'll say 10% more of the beneficial light plants use most. But it didn't say 10% more growth or 10% better growth. So, anyway, that's what chlorophyll A, the first form, looks like. The first one discovered. If you look at what hemoglobin's going to look like, which you probably won't do until we get to anatomy and physiology, but it looks like that, very similar, except with an iron in the middle. It's very green. So let's talk about the wavelengths of light. Light has different wavelengths, and the longer wavelengths, these are measured in, those are in nanometers, okay? No, they're not. Those are, yeah, nanometers, okay. And the long wavelengths, like red, have less energy. The short wavelengths have more energy. And you know that because, when you go out in the sun, what burns you? The ultraviolet. very high intensity of any radiation, this is part of the electromagnetic spectrum, will still burn you. You can buy infrared heat lamps that produce a ton of this, and it's absorbed by water. So here's that picture I just drew. I should have flipped mine around. Anyway, I did red first. I did low energy to high energy, but you can see it reflects green and absorbs blue violet and red orange. And there are accessory pigments that help them absorb other wavelengths. If I go back, you'll see there's some pigments that don't absorb red and blue, but start absorbing other colors like green, and they instead of reflecting green like chlorophyll, what do they reflect? Mainly orange and yellow. Some of them reflect red. A famous one is carotene, the pigment in carrots, but it's also, and those pigments are there all the time. They help fill in the spectrum and absorb more energy across the whole spectrum, but the chlorophyll predominates. At a certain point, when the temperature falls and the amount of sunlight per day falls, the cells stop making chlorophyll. And its job is so dangerous, absorbing energy, that it breaks down real fast. And if you don't continuously make chlorophyll, it goes away. Actually bleaches and goes away. And what you're left with are the other pigments. So when you stop making chlorophyll, the other pigments that are always there will show up. And those carotenoids, among other things, give you the red and yellow colors of the fall. One thing I like to do is take a coleus leaf and a spinach leaf and grind them up and do something called paper chromatography. We may do that one too. I forgot about that one. That would be a fun one to do. So, photosynthesis is an oxidation reduction reaction. That's where we have transfer of electrons from one to another. If you'll remember, oil rig, oxidation is loss, reduction is gain. And we're going to reduce our CO2 to carbohydrate, and that's going to involve transferring electrons. Where do those come from? They're going to come from water, okay? So, you can't have one without the other. So, in this thing, the water gets oxidized. It's called oxidation because, look, it's turned into oxygen. Before we even knew what electrons were, they still talked about oxidation because it gets more oxidized. And the CO2, if I convert carbohydrate to CO2, like in my respiration, that's oxidation. Because look, this carbon's mixed with some hydrogen and one oxygen for every carbon. But if I add more oxygen to it and pull the other things out, the ratio of oxygen gets much higher. Now it's 2 oxygens for every carbon. So I've oxidized that carbon in the glucose to carbon dioxide. So there's nothing magical about these terms. They've been used for a long time, and they just refer to how much oxygen is present. My water was a mixture of 2 hydrogens for 1 oxygen, and now it turns into just oxygen. So it's clearly oxidized. It does that, we now know, by loss of electrons. So, so that's going to gain. So, now the sun is the ultimate source of energy, but in our system, drop that toothpick that I brought for a lab in a demo. This sunlight is absorbed by the plants and converted to chemical energy stored in the bonds of sugars, either as starch or lipids or other things, because once you make sugar or glucose, you can make everything else in the cell. We can't, but many cells can. Simple E. coli bacterium can live on glucose alone and make every other protein, everything else in the cell that requires carbon, nucleic acids, lipids, everything, as long as it's given a little ammonium sulfate. It doesn't even need any amino acids, any nucleotides, anything. So, but all that glucose that every other plant, animal, and living organism on the earth depends on, all of that glucose comes by the reduction of carbon dioxide using the energy of the sun. And that's our trick, is to get from the energy contained in those photons, that's a chemical symbol, H nu, the energy of the photons. It's Planck's constant. It's a way to just say energy. Here, we just put a little sun there or something. So, star, what's a sun? And even a lot of the other things we think of as energy sources, not energy of the sun, really came from the sun. All the oil and gas is what? Broken down, fossilized life that floated to the bottom of the ocean, sediment grew over it, and after years, pressure converted it to oil and gas. And coal. I mean, you can mine coal, and that's the remains of ancient forests that fell down, got sedimented over. And you can, in some coal, you can crack it up, you can see plant leaves, you know, fossilized plant leaves and things still in it. What else? You know, just all of our solar energy now. It makes sense to use as much solar energy as possible. So, we're going to convert that solar energy to chemical energy, and we're going to store that chemical energy as a form of ATP. About 10 or 15 years ago, we stopped teaching respiration first and doing photosynthesis first. And it's kind of a chicken and the egg argument, but clearly, photosynthesis takes us to the sugar we're going to use when we do the reverse reaction. Okay, this is photosynthesis, this is respiration, where we, and plants do this too, that's a common misconception. Okay, plants do photosynthesis, animals do respiration. Plants make this sugar, but then they use respiration to do all the chemical conversions they need to make new cellular material, and also to extract energy from the stored ATP or the stored sugars, which get converted to ATP. So this sugar is oxidized, burned in the presence of oxygen, in a series of steps to produce carbon dioxide and water. And we already talked about ATP. So, and it's a high energy bond. There's actually two of them here. We have a good chemist, and that's not quite adenine, but that'll do. And attached to ribose, so ATP. When you break that bond, it releases tremendous amounts of energy, and it takes a large amount of energy to reattach that to make the ATP. And that's what we're going to do in both respiration and photosynthesis. So, two parts, reactions, two sets of reactions. The light dependent or just the light reactions obviously occur in the light. And this is where we absorb the sunlight and take that sunlight and use it to make the things we need to use later to turn CO2 into carbohydrate. Because this CO2 doesn't automatically combine with water, it takes a tremendous amount of energy. So our first step is capturing some of this energy and converting it to a usable form that will allow us to fix the carbon dioxide. It's called carbon fixation, and we're fixing the CO2 diffused in the air into the solid form of a sugar molecule. And we do that by taking the energy of the sun and its photons. Energy has both wave and particulate nature, but in the particulate nature, the photons, quanta of light, carry a specific energy and get absorbed. And I'll tell you what happens after they get absorbed. But ultimately, we are going to use that energy to move electrons around and make ATP and a carrier of electrons called NADPH. It's the first time we've seen it. We'll see it a lot in respiration as well. Nicotinamide adenine dinucleotide phosphate. And people didn't like that nicotinic acid because it sounds too much like nicotine. But this is a derivative of the vitamin called niacin. Okay? Vitamins are substances required in small amounts that serve as cofactors for enzymes and in catalysis. I'm not in the picture for you. Anyway, let me turn that over a little bit. So, if we have the light reactions, you want to guess what the other set of reactions are? The dark reactions. Or, more formally, the light-independent reactions, as opposed to light-dependent, and also known as the Calvin cycle, after Mr. Melvin Calvin. And is he Australian or from New Zealand? I want to say he's Australian. There's a picture of him in your book. He, and working with others, deduced the series of reactions that take carbon dioxide and fix it into chemicals, molecules that enter metabolism, or that are part of metabolism. Now, taking carbon dioxide and attaching it to another sugar, that requires energy because you're making a bond, you're making something, you need some energy. So you need to break a bond somewhere else, and you break the bonds of ATP to make ADP plus PI, release that energy, and you need some electrons because what are bonds? A sharing of electrons, and those are carried to you by this electron carrier called NADPH from the light reactions. So, in detail, in the thylakoid membrane, there are these reaction centers, these photosystems, that absorb photons of light, and it's called photosystem 1, or has a bunch of chlorophyll molecules. And it's amazing, you know, just almost picture, I can't draw that protoporphyrin 9 molecule. I could if you gave me enough time. But, you know, let's view them as these little discs. And they all line up, you know. And they form these huge arrays, literal arrays, just like, you know, a giant antenna dish array. And so you have these chlorophyll molecules, this is taking too much time, all arranged in something called a photosystem. Because what's going to happen is light is going to impinge on this photosystem. Light is going to fall on this photosystem and be absorbed by all those chlorophyll molecules. And one of them, after it absorbs enough energy, one of them is going to absorb so much energy, it's going to eject an electron. It's going to break a bond and eject an electron. A special reaction center electron, and this is too general. It's not giving me the specifics. Here we go. And so here come the photons, and they're all falling into all these chlorophyll molecules arrayed in the membrane in what's basically an antenna array. There's some accessory pigments mixed in, so if a photon of a wavelength of light that isn't absorbed by chlorophyll comes in, they'll absorb that, and they're absorbing all this stuff. You know, like laying out on the beach, getting all this energy. But at a certain point, they absorb so much energy that an electron is ejected. And these electron acceptors are proteins. It's called an ETS or ETC, electron transport system, or electron transport chain, because it follows a very specific path. Okay? Now, if you want to know the path, keep taking biology and chemistry and botany and biochemistry, and they'll teach you the names of these different molecules. Some are proteins. Some of them are small, ubiquitous molecules called ubiquinone, because it's ubiquitous and everywhere. Some are, you may have heard of CoQ. A lot of the weightlifters and people like to take CoQ. That's an electron transport molecule. But most of them are proteins, like iron sulfur proteins or heme proteins that have iron in them, or magnesium, or other things that allow metals, or, you know, what do we use to conduct electricity, or electrons, the flow of electrons in this room? Copper wires, hopefully. This wasn't built that brief period when we had aluminum wires. And we use metals because, we won't do the chemistry of metals, but they are very good at accepting and transferring electrons. And so this electron falls down back to ground state, because when you eject that electron, what are you left with? An electron deficient chlorophyll. It wants its electron back. There's two ways to get it. One is you can let it fall back down, and as it does, you can couple that to the production of ATP. Called cyclic electron flow, and we'll talk more about this when we talk about respiration and show you what can happen. And then the non-cyclic electron flow uses, this is more important, photosystem 2. It's a little difference between the two, but it's a series of chlorophyll molecules, an antenna array that absorbs sunlight. And it's only called photosystem 2 because it was the second one discovered. But it's the first one to act in these reactions. And photosystem 2 absorbs all this light and ejects an electron. And it falls back down the electron transport chain to where? To photosystem 1, which is also absorbing light and ejecting electrons. So let me show you this pathway, because it has to restore the electron a very special way. And this is shown here. So photosystem 2 ejects electrons. It falls back down to an electron transport chain to photosystem 1. Photosystem 1 doesn't want electrons, but it does, because at the same time that's absorbing sunlight, this is absorbing sunlight and ejecting electrons. So it says, okay, give me your electrons, because I have a void. I have a hole that I need to fill with electrons. So I'll take your electrons. But what does photosystem 2 do? Because it gave its electrons away, photosystem 1 has it, where does it get its electrons from? Where does it fill its void? You know, it ejected those electrons because it absorbed so much energy, those chemical bonds got so excited, those electrons were in so much motion, they could actually leave the molecule. And luckily there's someone hanging around who says, I'll take a high energy electron and I'll hand it off in successive lower and lower energy acceptors or energy levels back to photosystem 1. Well, photosystem 2 replaces its electrons with electrons from water. It splits water. It says, oh, look at all this water. We're in water. We're in an aqueous environment. I'll just take one of those water molecules. It's got electrons in its bonds. I'll pull those electrons out. That leaves some oxygen that as soon as one is made, another one comes by and they join together to form O2 because oxygen is naturally diatomic and likes to come together. It's a more stable environment. You never see single oxygen atoms. You get 1/2 of an O2. You don't get an O, you get 1/2 of an O2. And the next one over makes 1/2 of an O2. And they come together immediately. And you make H+. Protons. You know, a hydrogen atom, element number 1 is 1 proton, 1 electron. You pull the negatively charged electron out, you're left with its nucleus, its proton, H+. That's an acid. That's what we measure pH. So we split water, made an acid. Those electrons have to go somewhere. You can't have free electrons. If you have free electrons, or moving, if you have electrons in the air, you have a spark. You know, if you have them in a water or in a wire, you have a current. But they do fall back down. They're absorbed to fill the hole here, and this one went up here, and it transferred its electrons down an electron transport chain to this electron acceptor. It's actually something called nicotinamide adenine dinucleotide phosphate plus. It has a plus charge. And it takes an H plus and 2 electrons and forms NADPH. It carries electrons. Also carries away some of these. But this is called reduced NADPH. It's reduced because it's gained an electron. Reduction is gained, so it's reduced. So if you make ATP and NADPH, you can do anything because they're very high energy molecules. In this electron transport chain, we coupled the return of the electron to ground state. Where did that energy go? Well, some is lost as heat, but some of it's used to make ATP. So we absorbed it. Here's the bottom line. We absorbed energy from the sun in the light reactions, and we split water, forming oxygen and acid, H+. And we yanked those electrons out, and we transferred them to NADPH. Or we transferred them to NADP+ to form NADPH. The book isn't always clear on that point, but that's more detail than we need. So, where does the oxygen come from in photosynthesis? This is the classical question. I promise you you'll get it. We'll talk about it again before that test. But the oxygen, with every breath we take, came from water that was split by plants, or at least photosynthetic organisms. They're photosynthetic bacteria, for example, and protozoa. So there's this process called chemiosmosis. that powers our ATP synthesis. Osmosis is the movement of water across a semi-permeable membrane in response to a concentration gradient. I know, that was a week ago, and it would have been cooler if I had a lab with beakers and distilled water and stir plates and dialysis tubing and everything. And we have nothing. Well, we have some microscopes, but we don't even have microscope slides. All I can say is they're on order. A bunch of it did come in, but not everything I need to do any one experiment. A bunch of stuff for like 5 different experiments came in. So, chemiosmosis sounds really advanced, kind of is, but it's osmosis across the thylakoid membrane. Because remember, I have 2 areas in here, okay? I've got inside in the thylakoid and outside the thylakoid, but I'm still inside the chloroplast. So I have space here, I'll make it blue. And I have a space in here, which is inside. By the way, the fluid-filled contents of the chloroplast are called the stroma. That's not confusing because we have stoma or stomata. Stoma are the openings, stroma is the fluid-filled cytoplasm of the chloroplast. Anyway, when we make these H pluses, they're made asymmetrically, or we also call this vectorial ejection of the protons. Okay, so we split water and we made H plus. Big deal. Well, it is a big deal because all the H plus, I should have made these like pluses, is on the inside of the thylakoid membrane. Okay? And that sets up a charge and concentration gradient. There's an abundance of positive charge on one side. See all these pluses in here? And there's a dearth of, or a deficit of pluses on the outside. I wouldn't really call it minus, but relatively speaking, it certainly is minus on the outside. And if you have a membrane separating positive and negative charges, you just made a little battery. And you can use it to power things, and that's what happens. These protons want to go outside, but they can't because they're highly charged. They can't get through the membrane. But they run them through a protein called ATP synthase. It is, don't worry, I'm going to show you the types of questions we're going to ask. It's not going to go into this kind of detail. But it is an enzyme that synthesizes ATP. Okay? And so these naturally want to flow out because what does diffusion do? It goes from an area of high concentration to low concentration. It's blocked by the membrane, so it's hypertonic on the inside and hypotonic on the outside. And as these fall down and go from a high energy state, they're held in an artificially high concentration. That takes energy, but they release that energy and fall to the outside through this enzyme, and it couples that passage of the proton to make ATP. Amazing enzyme. It's shaped actually like a little mushroom. So you have photosystem 1 absorbing energy. Here it is, this little purple dot. And it splits oxygen. Oxygen can diffuse away. But the protons can't because they're too highly charged to get to the membrane. And it splits the water so that the protons end up on the inside. And it ejects that electron and sends it through an electron. This is photosystem 2, this is the first one. It sends that electron through an electron transport chain to photosystem 1, which is basically doing the same thing. Except, it's ejecting electrons and sending them through an electron transport chain, but it's not splitting water. It's getting its electron from the water through photosystem 2. It's just complicated things, I guess. So, those thylakoid membranes are there to increase the surface area. Stroma, not stoma. Stoma are the openings. Stroma means bed or mattress or something. But anyway, it's where the dark reactions or the light independent reactions occur. And the Calvin cycle, why is this term dark reaction a misleading term? Because it doesn't always happen in the dark, but there are plants that make ATP and NADPH during the day and fix CO2 at night. But the much better term is light independent reactions. And you're going to fix carbon dioxide. You notice we haven't even talked about carbon dioxide yet, just water and light. And there's an enzyme called rubisco. It is the most common protein on the face of the earth. There's more rubisco than any other protein, than all the muscle protein in all the cows in Texas. Because every plant uses rubisco. Its name is ribulose bisphosphate carboxylase, which is why I call it rubisco. Ribulose bisphosphate carboxylase, CO2ase. It fixes carbon dioxide, carboxylates it to a 5-carbon sugar called ribulose bisphosphate. You may remember RNA. That's ribose, 5-carbon sugar. Ribulose is a form of ribose. Okay? Very closely related. And that too should be subscript lowercase. Okay, so you take this 5-carbon sugar. So I don't lose you, I won't drop. And this, so I usually throw those away. You take this 5-carbon sugar, or needs treating, and you add a CO2 to it. So what's 5 plus 1? 5 plus 1? I'll pick on you. Marco, what's 5 plus 1? 6. So, and who has 6 carbons? Glucose. You make a 6-carbon sugar. I only wish it were that easy. You do make a 6-carbon sugar, but it's immediately split into 2 3-carbon sugars, glycerol derivatives. If you remember, we had triglycerides in the fats, and we're going to see this 3-carbon sugar, glyceraldehyde 3-phosphate. That's why we're going to just call it G3P. It's a glycerol. It's with an aldehyde group on it and a phosphate group on it. By the way, I got some glycerol. My glycerol came in. This didn't really come in. I just went to Walmart and bought some. But the glycerol's here. I'll let you taste it next time when everybody's here. That 6-carbon sugar is split into 2 3-carbon sugars. But once you have glyceraldehyde 3-phosphate, as you'll see next week, you can make anything. Okay. So, here's what happens. You take ribulose bisphosphate, 1, 2, 3, 4, 5, and 2 phosphates. When I first learned it, we called it RUDP, ribulose diphosphate, because it has two phosphates. But that doesn't say anything about how they're attached. It could be like ADP, where adenine is attached to one phosphate and a phosphate is attached to that, like the two phosphates together. Bis means on opposite sides or set apart, mirror image. So bisphosphate is a newer, better term. So talking about old people who call this the dark reactions too. This happening in the stroma, this ribulose bisphosphate with 5 has a carbon attached to it. It would be really cool if it were attached to the end, but it's not. Which is why it's broken into 2 3s. So if you could take this 5, 1, 2, 3, 4, 5, and you could attach it onto the end where the 2 phosphates are, on either end where the phosphates are, that would be cool. You'd make a 6. But you actually attach it, let's see, here. Okay? So that's why you split it into 2 3s. But once you have glyceraldehyde 3-phosphate, that can enter mainstream metabolism. In photosynthesis in the Calvin cycle, we make 3-phosphoglyceric acid, 3-PGA, also known as 3-phosphoglycerate. That's just the salt form of the acid. And then ATP is used to, see, if this builds up in balancing reactions, this will feed back and stop this. So you need to swoop this away. And one way to swoop it away is convert it into something else. So its concentration drops to 0. And it's naturally, the enzyme will continue to catalyze this one at a time. And so we'll add a carbon, 1 CO2 to this to make a 6-carbon version of this that's immediately split into 3. And as soon as it's split into 3, it's phosphorylated to get another phosphate. We start out with 2 phosphates, but we split into 2 so that each of the 3s only have 1. Then we add another phosphate. That effectively removes it from the equation for that reaction and forces that reaction to flow toward production. Forget the energetics. Anyway, we make this compound that gets gets reduced. It's glycerate, the glyceric acid. It goes from the acid to the aldehyde, and then from the aldehyde to the alcohol. That means it's gathering electrons, being reduced, and also being less oxidized, less oxygenated. So it's also picking up some H's. So I'm not sure using little spheres is any better than actually using the structure. But this glyceraldehyde 3-phosphate, who we'll meet again next week, goes in several steps through something called the pentose, derivative of the pentose phosphate pathway. That's the pathway you use, everybody uses to make 5-carbon sugars, to make DNA and RNA. Everybody has it. Everybody can make the precursors for DNA and RNA. You don't need adenine, thymine, cytosine, or guanine in your diet. You need 8 amino acids. You need omega-3 fatty acids in your diet. You need all kinds of things in your diet, but you don't need anything to make DNA. DNA is too important to not be able to make it, including the 5-carbon sugars, ribose and deoxyribose. Well, you can use that pathway, or parts of it, to make ribulose, as well as ribose and deoxyribose. And so these plants use that pathway to convert the glyceraldehyde 3-phosphate to ribulose bisphosphate. And here's that rubisco, so they are using my terminology again, at least. The point is, when I did this with one CO2, I took a 5 carbon and made a 6 carbon that immediately broke into 2 3s. And then those got converted to more and more reduced 3 carbons. But I have 2 of them, and one thing I can do is, when I get a bunch of glyceraldehyde 3-phosphate, remember I kept saying you can go anywhere. Here's where you can go. Let me show you 2 places you can go. You can combine 2 of them together to make glucose, and that's exactly what you do. The 2 glyceraldehyde 3-phosphates come together to make glucose. We're going to split them apart next week to get energy from them. But you can put them together to make glucose. And you're making lots of them. And you can take 5 of them, 5 3s, to make 3 5s. You know, you've put 2 3s together, you make a 6. And then you put another 3 together, and you don't make a 9, you make a 5 and a 4. And then the 4 you combine with the 3 to make a 7 carbon sugar. And then you add another 3 to that, and you don't make a 10 carbon sugar, you make 2 5s. So it looks like a lot's going on, but what we're really doing is spinning out 5s, 5 carbon sugars, so that we can fix CO2 to make 3 carbon sugars that we can go on to make 6 carbon sugar that we love, glucose, or fructose, which is 1 step away, or galactose, which is 1 step away. So that's it. So this is called C3 fixation. There are plants that do. Most plants are C3 and use the Calvin cycle. There are other plants like cacti that don't make phosphoglyceraldehyde, and they have different we're going to talk about photorespiration. It's a way to quickly make ATP. But some plants called C4 plants, instead of using C3 and C5, they use C4, you know, and CAM plants. So you can look at this. There's a little tiny bit. C4 plants are some grasses and corn and sugarcane, which are important around here. The CAM plants are like the cacti, and they have slightly different carbon fixation. They attach it to a C4 compound, malic acid, malate here, that, gives you a little bit that you can exploit too. So that's why you can, because they do a different type of photosynthesis, cactus and succulents, and these are the ones that have the true dark reaction. Stomata only open during the night. Why is that? If you're living in a desert environment and you open your stomata so oxygen and CO2 can get in, water can get out, because what's the water going to do? Move from an area of high concentration to low concentration. You have dry desert air, you lose all your water. So you open up your stomata at night when it's cooler, and you lose less water. It's an interesting adaptation. The dark reactions truly are dark. And the only thing I want to say about these C3 and CAM plants is because they have different mechanisms of photosynthesis, you can design herbicides and pesticides that attack them specifically. And that's important if you are a sugarcane or corn grower. You know, you can use different herbicides that will kill them, or won't kill them, but will kill your neighbor's soybeans. You may have heard of some of the controversies or arguments going on in Arkansas over doing these. If I may, I'm going to talk about the test. So that's it, we're done. I said 6 o'clock, give me 20 minutes to talk about the test. The classroom next door has a trash can near the teacher's desk, but oh well, I don't need this. That was pretty heavy aspects of photosynthesis. So I'm gonna sit here because I can control my computer better and talk at the same time. You can't see me on the camera, too bad. So let me show you photosynthesis utilizes the entire electromagnetic spectrum. That's a terrible question because you might remember, oh, he said that they don't absorb green light, they reflect it. And they really only use visible light, but not all of it. So I would reword that. Which of the following is only minimally absorbed? You need to know that. Green light is reflected, not absorbed. The end products of photosynthesis. So these have been posted. We'll talk about them again before we give you a test over them. But I want to talk to you about the other things on your blackboard. So the reactants, the things that react, include all the following. Glucose is a product, not a reactant. And this is the classic question. These were actually pre-test questions. There are, at CARC, I had to show that you learned stuff. And so every class had to give a pre-test at the beginning of the year that included questions from the final so that I could show you learned something. And that's easy to show, but we had to prove it. But that's why these are called pre-questions, because, you know, the oxygen comes from the water. And we'll talk about these. So these get reported in these 20 questions, but that's the level. A plant can use glyceraldehyde 3-phosphate to make all of the following except actually it can make all of those. That's a terrible question. The energy needed come from NADPH and ATP. So I want to go over the ones for this next test though, okay? I won't ask the C4 and the CAM plant questions, and why they grow better when it's, you know, hot and dry, whereas others grow better in the moisture and everything. So, which is not a product of the light dependent. Glucose, the sugar gets made in the light independent reactions. So, you can see these don't go into too much detail about the thylakoids and the way the chemiosmotic gradient is set up. So, let me go back and talk about our next test. 1, 2, 3, 4. So if you look here, I have several things. Here is a test I gave a long time ago. And here are some more test questions I can use, because I haven't made it. But let me put these out and show you what we have here. So, these are my quick and dirty notes about cells, cell theory, the organelles, the membrane, the cell wall, the cytoplasm, the ER, the ribosomes, Golgi. Oops, I could have sworn I formatted everything. I hate those. Oh, I guess that was okay. There wasn't any description about the Golgi. It's just flattened stacks of ER. So, you know, this is just a list of everything, and I ruined it, so I'm going to close it and not save it. very quick, if you don't have much time and you don't read the chapter, that's what you want. The other thing I'd like you to look at here is, of course, this outline produced by the, oops, that's the notes, sorry. This outline produced by your publisher, which is much more detailed, okay? I would say Schleiden and Swan. Virchow was really working with viruses. I never really looked through this. I'm not sure. It talks about viewing cells, prokaryotic cells versus eukaryotic cells with a true nucleus. That's the difference, the true nucleus. It goes through all of the different parts of the cell, lysosomes filled with powerful digestive enzymes that are highly acidic. They fuse with food vesicles and digest the contents of the vesicle. White blood cells are filled with lysosomes. Also, we are not going to discuss it, but there's something called apoptosis. It's programmed cell death, where cells die off after a certain amount of time or a certain number of divisions, and the lysosomes digest them from the inside out, so there's some control over that. Boy, lots of good stuff in here, including stuff I didn't talk about. So remember two things. If I talked about it, it's not in the book, I'm not going to test over it. If you're taking careful notes or paying careful attention and can remember what I said. Because that's not fair. You know, sometimes people ask me questions and I go off on a tangent. Today I talked about the dorsolateral prefrontal cortex, which is neuropsychology, neurobiology. So obviously that's not there. You got to be able to look it up so that you can figure out, what did he say? What was he talking about? And go to the book and look it up, because you're too shy to ask me during class. So, and ask questions. And don't y'all go, ugh, when somebody asks a question and keeps us here 2 minutes longer. And the other thing is, if it's in the book, and there's a lot of stuff in your book, because it's encyclopedic. Look how big and thick and fat that book is. And I didn't talk about it. I don't think it's important. That's me testing your reading capability. Did you go read the book? Okay? Which is why I like my little outlines. But the other thing is, I'm lazy. I close this. Did I close that down? I don't know what's going on here. Let me just open it up again. So here's sample test. Hello, here it is. Okay, so these are sample test questions from the McGraw-Hill website, from their test bank, and here's an old biology test from 2018. Now this was short. That year I gave a test every week. I like it, but anyway. So I had a picture from the book at the time that asked, what are these structures? You know. So, but they weren't just, what is A? What structure is found in a plant cell but not an animal cell?
Cell wall.
You know, that would be if I had a picture of a plant cell. So that you would say chloroplast or cell wall. Or which structure is found in an animal cell but not a plant cell? And that would be centrioles. But I didn't emphasize that, so we'll emphasize that when we get to cell division. So sometimes they're functional. So this structure moves materials between the organelles of the endomembrane system, or centrioles are made up of, but basically it was
label the indicated structure in this diagram, and they had to fill in the blank. Oh, I just put this picture up there so they could see. Yeah, label the indicated structures diagram in the spaces provided on the last page. So, these are okay questions, and there's a picture of a cell. I'll use this one or the one from your book. I have access to both of them. So let's talk about questions, because this is, there are only so many ways to ask what does a mitochondria do, or which organelles have two membranes, or which organelle contains enzymes that break things down, hydrolytic enzymes. Hydrolysis was one of the questions from the previous test. So, smallest living unit is a cell. I should make you look these up, but I'll just go through them real fast. Protein synthesis in bacterial cells, and every cell is performed by ribosomes. I didn't make a big distinction, but bacterial ribosomes are smaller. We call them 70S. The S is a measure of how fast they go through a gradient. So the smaller the number, the smaller they are. And eukaryotes have 80S ribosomes, but the ribosomes in the chloroplast have 70S ribosomes, just like bacteria. The outermost boundary of prokaryotic cells is their capsule. That's not a choice. The outer membrane, that's not a choice. The cell wall. They have a inner and outer membrane, and they may have a capsule as well. Sperm cells move by means of flagella. Lung cells move by means of cilia, for example. Pseudopods, false feet, are how amoeboid cells like white blood cells move. I'm just looking, because sometimes I'll kind of change these up. Skeletal cells have a large number of mitochondria because of their high demand for energy. So this is a good example of, you didn't talk about skeletal cells, but this is like a secondary, question. I didn't say what organelle produces energy. I said, think about it. If you were a cell that required lots of energy, you would have a lot of this organelle. White blood cells that engulf bacteria and digest them would have a lot of lysosomes. So, antibody producing cells that prepare lots of protein, antibodies for transport and export out of the cell would have lots of what? Golgi apparatus. Okay. I didn't talk about how the endoplasmic reticulum is responsible for, we used to say drug detoxification because we don't do these experiments anymore. If we gave a rabbit lots of barbital, it used to be the most prescribed drug. It's a barbiturate. Barbital, barbituric acid, but it calms people down or does other things. But if you injected an animal with barbital, can't do this on humans, and then slice their liver open, look at their liver cells, the endoplasmic reticulum proliferates because the enzymes involved in drug detoxification frequently don't function in water or around too much oxygen, so they happen inside lots of membrane, and membrane proliferates in liver cells of people who are exposed to environmental contaminants or drugs or things like that. And I just said it's the plumbing system of the cell and used for transport of things throughout the cell and the site of membrane synthesis, because all of the organelles pinch off and start out as endoplasmic reticulum. Smallest living unit is a cell that's part of cell theory. They repeat these in the regular ones as opposed to pretest. All cells contain the following structures except everybody has ribosomes, everybody has cytoplasm, everybody has a plasma membrane, and prokaryotes, bacteria and archaebacteria, we have to distinguish between them now, do not have a nucleus. They have a circular DNA chromosome. That's for protection because they tend to get broken down from the end. And our DNA is so important, we package it away inside a double membrane organelle and sequester it. And then when we need to make protein from those instructions, we say, I'm not gonna let you see it or use it, but I'll make you a copy. And we make an mRNA copy. I mean, think about, cells do things for very logical reasons. Okay, so this, we didn't talk about until this week. The stacks of membrane discs containing chlorophyll and chloroplast are the grana, which is actually, someone thought those discs looked like granaries or grain storage towers, which is kind of appropriate considering this is where the sugars are gonna be, well, it's in the stroma and the dark reactions, but they didn't know any better. You know, in that chloroplast is where the sugars are made. They get turned into starch that we store as corn or rice or beans or whatever. So grana is short for granary or a Latin derivation of it. Large storage structures found in the center of plant cells are central vacuoles. You'll color these. You know, when you go to color these coloring sheets, you'll see all of these. Ooh, I love this. Which of the following statements is false? This is like asking 4 questions at once. Some cells are large enough to be seen by the naked eye. So think of a chicken egg. It's a single cell. The human cell is about 100 microns big. That sounds really small, but that's 1/10 of a millimeter. If you line 10 of them up, they would be the distance between the lines on a millimeter ruler. Or if you line 3 of them up, put them 3 human egg cells, it is inside the visual range. 3 of them together would be the size of a period, the end of the sentence in your book. So you can see, you never see it, unless you're maybe working in a fertility lab or something. But, and you have it in a petri dish with nothing else there and lots of light and a nice white background, but it can be seen by the naked eye, but in general, not. Cells with infoldings and outfoldings of their plasma membrane are able to move molecules across the membrane more efficiently than cells with no foldings. And that's true, we're increasing surface area. That's a central theme. You know, that's why I have all this thylakoid membrane. Very slow moving algae that are very inefficient have just big hollow chloroplast with one membrane then another membrane. You know, they're not as efficient as, I mean, just wait until they plant the corn. Have you ever driven by a cornfield and like a day later, it's like, they say, and I've never been there, anybody live in the country? We all live in the country. They say you can hear the corn growing at night when it's quiet. I don't believe it, but I haven't cared enough to go out in the middle of a cornfield in the middle of the night. But maybe we'll have a field trip. Anyway, all cells are shaped like cubes are round. That's clearly false. Every shape in the world, and eukaryotic cells, the nucleus controls the activities of the cell. That's a little trite, but okay. Eukaryotic cells are different from prokaryotics in the following ways. Everybody has cytoplasm, everybody has a cell membrane, everybody has DNA as their genetic material, but eukaryotic cells have a nucleus and a variety of organelles. You know, remember I showed you that movie of the contractile vacuole that fills up with water and goes and squeezes it all out. You only see those in freshwater species. Another thing that some cells do, and I ordered it and it was supposed to come in today, is some Alodia canadensis, but I found that's against the law because it's an invasive species. I can't buy it anymore. But I found a different species of Alodia or Alodia. I don't know how to pronounce it. I've heard it both ways by botanists. It's also known as Anacris now. That plant, it's a common pond plant or aquarium plant. The leaves are just a few layers of cells thick, so you can just basically put the leaf under a microscope, on a slide under a cover slip. When you look at the cell, there are these big, beautiful square boxes. You can see the chloroplast moving around and tumbling. It's called chloroplast streaming. And it happens as soon as you turn on the light. If you turn off the light or shut the light down, you can see them slow. So I'll bring that in. And that's a way to what? Increase surface area. You know? Oh, wait. You know, I'm on the bottom. The sun's up here, the light's up here. I'm on the bottom of the chloroplast. Not the cell, not the leaf. I'm on the bottom of the chloroplast. Turn over. You know, so I can do it. And then he goes to the next guy. Hey, wait, you're up there. Move over. I'm going to move over here. It's wonderfully logical, and it's in lots of plants. While we're talking about streaming in Elodia. And it, you know, it's something we can view very easily. Look at those. It's so cool seeing them just kind of marching around the outside of the sphere, the cell. See, those are big box-like structures. Why aren't they marching around in the middle? You know, these cells are so cool that, you know, you think, well, you want to increase surface area. Why don't you? This wouldn't be in this slide. That's not also like these. So cool. Why don't you have more chloroplasts in the middle of the cell? Because that's that big vacuole controlling fluid pressure and osmotic pressure. Because these live not in seawater that's isotonic, but in pond water, and the water rushes in. So there isn't a lot of cytoplasm because really most of this cell is vacuole. But in some of them, you can see that little shadow of a round thing? That's the nucleus. It's off to the side. But, you know, this is nothing magical about this. We can do this in the lab, and we'll do it in the lab. It's so cool. Thank you, Mr. Ralph Grimm, whoever you are. I will go back and follow you. So, almost done. So, which of these organisms contain hydrolytic enzymes? Of course, that's lysosomes. Synthesis of lipids occurs in the smooth ER, and membrane-bound proteins in the ribosomes attached to the rough ER. And what is it that makes the surface of the rough ER run? Ribosomes that are attached there. 9+2 array refers to microtubules in cilia. And as cells increase in size, the volume increases faster than the surface area because it increases with the cube and the surface area increases with the square. I don't know where the answer is. There will be a greater increase in volume than in the surface area. There won't be enough surface of the cell to allow diffusion into the cell so the cell can carry out metabolism. Okay, I didn't talk about signal recognition proteins, but when a protein first starts being made, if it's sticky and hydrophobic, it will stick into the membrane of the ER. The signal recognition protein binds first to the signal peptide and the polypeptide being synthesized into a receptor on the ER, which opens the channel. You know, do y'all know test-taking strategies? We're going to talk a little bit more about this next time when everybody's here, and a little bit more about how to study. If there's a big, long answer, it's probably the right one, because the person writing the question, that's me, wants to be precise and make sure if there's an exception that it's covered or that we really get it right, you know. So 9 times out of 10, you're going to pick the longest answer in the ACT. The ACT knows these tricks, so they tend to work around them too. But, you know, when you see a big, long description like this, they had to do it to get it right. The enfoldings in the inner membrane of the mitochondria provide increase in surface area and important ATP production and cellular uptake. Name these structures. Okay, you won't get that because I just talked about mitochondria. We'll talk about them later. They're called Christi. There they are. The fluid contents of the mitochondria are the matrix, just like the fluid contents of the chloroplast or the stroma, as opposed to the stoma, which are the openings in the plant cells. Which of the following will not be found in abundance in an insulin-producing pancreatic islet cell? So they would have lots of ribosomes because, okay, you don't know this. Insulin is a protein. To make a protein, what do you need? You need ribosomes. And if you're going to secrete that protein hormone, what do you need? You need Golgi apparatus, and you need those ribosomes to be attached to the ER so that it's called pre-proinsulin. It's a precursor to insulin. That insulin is produced, the pre-proinsulin, is put into the lumen or the inside or the cavity of the endoplasmic reticulum so that when it pinches off from the Golgi, it's inside there as opposed to the cytoplasm. So that's a lot of reasoning, but you can always ask me to reword things. Debris that is trapped in mucus and moved away from the lungs toward the throat by cilia. We have ciliated cells. They're constantly pushing material the right direction. If you stand upside down, they'll still brush it up, or in this case, brush it down until it gets the back of your throat, touches the back of your throat, and makes you do that and swallow it. A dark staining spherical body found in the nucleus that produces ribosomal subunits is a nucleolus. And the endosymbiotic theory explains the origins of chloroplasts and mitochondria. And then I did put some of the questions from chapter 5 on, but don't go into the heavy stuff about osmosis and diffusion. It's just that some of this is also found back in chapter 4 when they briefly discuss membranes. So that's in your course. I put the stuff for tonight in here, including multiple copies of this. Here. I don't know why they got 2 copies of that. Where is it? Here. No, that's not what I wanted. Oh. Did that remove it? Nope. Mark for removal, not submit. Okay. Anyway, that's it. Good night. See you tomorrow. We'll have a test over chapter 4, lightly chapter 5, cells and membranes. And we'll do respiration afterwards. And I will stop this recording. I might break this into 2 parts. The talking about our test and the talking about photosynthesis.